In this video, we look at Key Area 4, Part B, Genetic Screening and Counselling. You should already know about alleles, dominance, recessive, homozygous, heterozygous, genotype and phenotype. We'll talk about the terms autosomes and autosomal chromosomes, sex-linked in terms of inheritance, and incomplete dominance. So patterns of inheritance can be determined by genetic screening and the use of what are called pedigrees. Counselling can then be offered to parents to allow them to make decisions as to how the pregnancy should proceed. If one or both parents know that they have a particular genetic condition or abnormality, you can work out using pedigree charts and Punnett squares how likely it is that you will pass that on to the next generation. So, what are the sex chromosomes in humans? And have we think about that for just now? Okay, so we have 23 different types of chromosomes. Chromosomes 1 to 22 are all sections of DNA that have different genes. All the genes that are on those chromosomes are autosomal genes because chromosomes 1 through 22 are your autosomes or your autosomal chromosomes. X and Y are your sex chromosomes. XX makes you natally female and XY makes you natally male. Family trees look a lot like this. We can use these, as I said, to describe from phenotype only what the genotypes of those members of the family must have been. So we know from National 5 that if two individuals with the same phenotype are able to produce an individual of a different phenotype, then it must have been a recessive condition that was hiding in their genotype. We can use our knowledge from National 5 in the higher course, and actually there's very little extra, really sex linkage and the word autosome are only really what we're adding. So in a cross, so when we have two individuals who we know their genotype and we mate them to produce offspring, they are the parental generation. They produce the F1 or first filial generation, which again we outcross to people who are not their siblings usually. And you produce from the F1 what's called the F2. So the parental produce the F1 produce the F2. And then you have F3, F4, F5. So a term here that's not really in the course, it's in the National 4 course, but at some point we keep using it in the PowerPoints. True breeding just means the same as homozygous. So a true breeding white mouse must be homozygous. True breeding grey must be homozygous. Now, because between the two of them, they produce only grey offspring, which we then incross or incest mate, white must be homozygous recessive and grey must be homozygous dominant. Because of that, grey can only pass on the dominant allele and white can only pass on the recessive allele because that's all it has. So both of these will be heterozygotes. If we cross them together, you can see we have three grey looking mice. It's likely that this one would be homozygous dominant and these two would be heterozygotes just by the Punnett square. And this one, the two recessive alleles, 1 in 4 times will come together. If we had fruit flies with normal wings and vestigial wings, normal wings is the dominant allele. So the allele is the forms of the gene. If we looked at the gene, so the section of chromosome that determines hair color, we could have alleles such as black, brown, blonde, etc. These are the alleles. One of these alleles will be dominant to the other. Normal wing is dominant over vestigial wing. Vestigial wing is said to be recessive to normal. If we true breed crossed normal winged fly, so capital N, with true breeding vestigial, so small n, what's the possibility? Is it possible to work out what the genotype of the offspring will be? You can actually 100% of the time say that the normal one will pass on only the normal allele, and the vestigial one in this case, because they're both true breeding, will pass on the vestigial. So all of their children, so from the parental, all in the F1 will be a heterozygote, so capital N, small n. And because of that, because capital N is dominant and it's normal winged, all of the F1 will be phenotypically normal winged. And we can see that here, the cross, the allele, so the gametes that it's able to produce is just the N, just the small n, so all will be that. The genotype is big N, small n, and phenotype, so the appearance is normal winged. Phenotype isn't always appearance. If you think about the gene that allows you to produce insulin, the phenotype for that gene, there's an allele which produces insulin and there's an allele for not producing insulin. So it's not always something physical that you can see. We could put these in a Punnett squares. Punnett squares are useful when you can't just do the genetics off the top of your head. The more you practice this, the more likely you are to be able to just do it on the top of your head. So 50% of the time, the big N will be put forward by this parent, and 50% of the time, the small n will be put forward. And the same here, same here. So if this one puts down its big N and so does this one, that will happen 1 in every 4 times. So 1 in every 4 times, you will get big N, big N. 1 in every 4 times, you got small n, and 50% of the time, one of them will put in a dominant allele and one will put in a recessive, because there's 2 different ways that can happen. By probability, you would assume that if these 2 F1 parents were to mate 4 times, they would produce 3 that were normal winged. Of those 3, 1 was a homozygote and 2 were heterozygotes, and 1 of the 4 would be small winged vestigial. This is showing it normal phenotype, normal phenotype, normal phenotype, vestigial. So the genotypes are here, and these would be the phenotypes for them. It's a 3-to-1 ratio. Quite often they will ask you what the ratio is. That's not necessarily what would happen. King Henry VIII had multiple wives. He never once had a son. He kept mating because of what we will look at with sex chromosomes. Females can only put in X chromosomes because they only have X chromosomes. So every time Henry VIII's wives would put in a chromosome, it would be X for sex. And Henry VIII was the one who kept putting the Y chromosomes in. Males decide the gender of the child because they have an X and a Y. So only the male, we'll see when we look at sex-linked genetics, can decide if the child is going to be male or female. So it was his fault, not theirs. So sex is random. That's the answer to this. It's a random chance of if every time you're going to pick this allele or that one, you would assume in probability that you would get this distribution, but not necessarily. So family trees. Tongue rolling is an inherited single gene characteristic, which means that it's only controlled by one gene. Skin color, for example, is controlled by multiple genes, which is why you get different degradations of melanism. So the tongue rolling allele is dominant over not tongue rolling. So this is what we would say here. Big T is tongue rolling, small t is not tongue rolling. We've coloured in some circles here to involve the sex of the individual. It doesn't really matter for what's called autosomal characteristics like these, but we'll still involve it. So the blue colour will be tongue rolling and the white colour will be not tongue rolling. Circles will be females, squares will be males. So in this one here, you can actually guess your way through a lot of these genotypes. We've already said that if 2 individuals of one colour are able to produce an individual of a different colour, they must be heterozygotes, and the colour they are, like we see here, must be dominant. So we already knew that tongue rolling was dominant, but now we've just had that confirmed. So these 2 must be heterozygotes because they are capable of having one phenotype and producing an offspring of a different phenotype. The first thing you can actually do is go through, and every time you see non-tongue rolling as a phenotype, that must be a homozygous recessive individual. So that has to be small t, 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 small t. So you can write that on your bit of paper. It often makes it easier. It's the first thing we do. And then you can go around looking for little tricks like that. So these must be heterozygotes. For these 2 to be able to produce this individual here, this one must be hiding a small t, because if he passed his big T down to this individual, this female here, she would be a blue circle. So this one here, there we go, must be big T, small t. This one here, because it's the child of these 2, again, it gets big T from the father, and the mother can only give little t. So every one of their progeny, their children, must have at least one little t, which makes that a heterozygote and that a heterozygote. This one here again must be a heterozygote because it's able to produce this, and then you can follow this through. These 2 will have a big T from their mother and a small t from their father. Here we actually don't know if this one is big T, because that's possible, or big T, small t, because that's possible. We just know it's not small t, because it's not white. This one here, because all of these get the little t from their mother, technically, as we said, observed number of offspring with one phenotype and actual number predicted are quite often different. It's possible for this one here to be a big T small t or a big T. Every one of these 4 chances it is chucked out as big T, but it's possible that it's a heterozygote instead. And we don't know for this one. So these are some of the terms that we should know. Autosomal is just the what we did at National 5. It's using the 1 to 22 chromosomes of the body. You don't need to worry about if the individual you're talking about is male or female when you're looking at autosomal, because males and females have the same chromosomes. Recessive, we've talked about. Dominant, we've talked about. Homozygous and heterozygous, we've talked about. Incomplete dominance and sex linked is slightly different.